Hello, our chapter is 4, Technology Platforms for Program Pharmacology. We sometimes use the pharmacological interventions for animal some behavior change and neuronal modulation. However, there are limitations of pharmacological injection because we have to do anesthetize or fix the animals for injections. This unnatural state, such as anesthetizing, may affect the neuronal circuitry and behavior of mouse. That was our major interesting point. Nowadays, there are few techniques can avoid or minimize these limitations. The animals can be injected when free-moving state. These techniques also have precise temporal control of multiple drugs. Let's see what is some techniques. The first one is the product combining microfluidics and microelectrodes. We can deliver the fluid up to 3 different fluids to our target lesions, and through the 5 microfluidic channels. So we can modulate the flow rate levels to the 5 levels. We can block each of the 5 channels, then the flow rate is decreased. And this light image is the cross-sectional image. So we can also measure the local field potential through this 16 electrode, microelectrode, and this microelectrode is around the outlet of the microfluidics. And the other is the fiber type microfluidics interface that was combining products of the microfluidics with the optical fiber. So this image is the drug is delivered through the fibers and this core and cladding. So this core cladding this is the optical fiber, and cladding includes the 2 microfluidic channels, and there is 6 electrodes. So this is the whole image. So we can connect the laser to the pedals, and the wire is the load of the ground electrodes, and this tube is connected to this microfluid, and fiber is go through the brain. In this optical fiber complex type is enable for us the 2 steps to the 1 steps. The 2 step is in the optical stimulation, we have to inject the virus, such as some channel rhodopsin virus, and also implant the optical fiber. But this interface can summarize these 2 steps to the 1 steps. And the final one is the fully integrated and the self-contained device that eliminates the requirements for physical scissoring. So we can see this designed device is no connection between some other instruments. So we can literally say this is a really free moving mouse. So the left one is the drug and light is the light delivery. So, and both of them can be occurred simultaneously.
Then how are those drug delivery platforms used for neuroscience research? First, I'll talk about silicon shanks. This platform is used to demonstrate hippocampal and CA3 and CA1 connectivity in vivo. This is the illustration of 4 shanks, and the drugs used are CNQX and AP5, which is neurotransmitter receptor antagonist. When we deliver the drug as CA3, they are delivered to CA1, and it would block the synaptic transmission. This is the graph showing neuronal activities at S1 to S4. This blue mark indicates the light onset, and the light activates CA3. Before injecting the drug, neuronal activity pattern is identical among S1 and S4. After injecting drug, the neural activity at CA1 is decayed. So it enables the study of long-range neuronal circuits. Next is the polymeric fiber. This can be used to explore behaviors in unanesthetized and tethered animals. And this can be used, so it was used to demonstrate the BLA neuronal projection to VHPC in the context of fear and anxiety. Anxiety could be measured by open field test, and if the time spent at center is less, it's defined as anxiety. This graph, um, before the graph, uh, CHR2 carrying virus was injected to BLA, and VHPC was activated by light. And this graph shows the time spent in the center of open field. And before injecting CNQX, channel rhodopsin 2 mice spends lower time at center. However, after injecting CNQX, which means this pathway is blocked, there was no significant difference between control and CHR2. Next is wireless drug delivery, which is fully wireless and tethered-free system. This, as the previous studies showed that unilateral opioid stimulation at VTA causes an increase in locomotor and training behavior. They either delivered artificial agonist cerebral spinal fluid as a vehicle control or mu opioid receptor agonist, which is DEMGO, into mice wirelessly. This is the heat map of at open field chamber. And as you can see, DEMGO delivered mice showed increased locomotor behavior. So this system operates with a remotely controlled interface to independently infuse multiple fluids in various environments. Lastly, it is also the wireless technique, and this probe enables the bidirectional wireless communication for real time. By using this model, they tested whether selective activation of the VGAT pathway would produce a specific preference. This is a heat map of 3 cases. First, when no stimulation is given, mouse shows no preference. When photostimulation is given at upper chamber, a mouse shows preference to stay at upper chamber. Then gabazin is microinfused into LH and blocks the pathway. So gabazin infused mice loses preference even when photostimulated at upper chamber. This model is effective since we can observe the behavior at 3 different environments on real time. By today's presentation, we learned about technology platforms for programmed pharmacology and how those technologies were applied in neuroscience research. Thank you.